Good evening and welcome to this regular meeting of the Entomological Society of Washington. To start, Secretary Gary Havel, could you please read the minutes from the last meeting?
Yeah. The 1258th regular meeting of the Entomological Society of Washington was held on the 5th of February 2026 in room WG13 of the National Museum of Natural History with President Talita Samoas, bringing the meeting to order at 7 p.m. Present were 13 members, 3 guests, and 18 virtual participants. Recording Secretary Gary Havel read the minutes from the January meeting with approval by members at the meeting. Membership and Communications Secretary Matt Buffington noted that there are currently 253 members in the Society with the following 8 new applicants: Benjamin Bergender, Karen Berghart, Clayton Ziemke, Mark Hoddle, Courtney Painter, James Woolley, Roy Beckenmeyer, and Raymond Sherbull. For new business, Matt Buffington displayed a large singly bound volume that included all of C.B. Riley's reports. Matt had received this treasure from one of his college friends and noted that the volume could be examined in his office rather than send it around the room. President Samoas noted that the Society currently has one opening in a section of honorary members and explained that the Executive Committee had agreed that Warren Steiner would be a proper addition to that grouping. Warren received a unanimous yes vote from attending members and responded virtually later in the evening with a heartfelt and thankful message for this honor. Program Chair Renan Carreño arranged the speaker of the evening, Zachary Griebenau, who is currently a postdoctoral fellow with the Systematic Lab of the USDA. Dr. Griebenau was introduced by Matt Buffington. The title of the presentation was An Interpretive Systematic Revision of the subfamily Leptonillinae. This subfamily has many distinctions as they are subterranean and rarely seen, and almost never observed alive. They emerged in the early Cenozoic. They are found only in the Eastern Hemisphere, and their genitalia are often bizarre. In 1925, Bruez was confused by their taxonomy. Only in recent years have there been advances in taxonomic placement. 2 species are known only from single specimens. The anatomy of male genitalia is weird and not yet described in literature. Due to the rarity of specimens, researchers have used specialized computerization. Collections of these ants would be helpful to taxonomic studies. They live in soil, and sifting soil can be productive. Dr. Griebenau noted that he has never personally collected any specimens of this subfamily. After much post-presentation discussion, the meeting was adjourned at 8:25 p.m.
Thank you, Secretary.
Sure.
Does anybody has additions or corrections to the minutes as read? I'm not sure, but maybe you said that what Matt brought was new business and it was note and exhibit.
Yeah, I'll take a look. Yeah.
Yeah.
Okay.
Can I have a motion to have the minutes approved as read? Second. Thank you. All in favor? Thank you. The minutes are approved. Secretary Gabel would also has an announcement.
Yeah, there's been a couple of deaths in the entomology group here. People who have worked some time ago. I'm not certain that they were members of the society. However, we'll just mention the 2 briefly. Society member Paul Goldstein recently announced the passing of Robert W. Poole. Bob was an enormously productive scientist with the Systematic Entomology Lab for 15 years in the 80s and 90s. His contributions to primary systematics research were many, but he is perhaps known most widely for his 1989 catalog of world noctuids. later for the development of Nomina insecta nearctica. Bob and his wife, Patricia Gentile Poole, formerly of SI Entomology, moved from the DMV to her native Argentina in 2016. He was a valued collaborator and mentor to many students and postdocs, and he maintained a presence at the museum long after his normal departure in 1995. He will be missed. Word has been received of the recent passing of former museum specialist Beth Norden. She was an assistant to Dr. Carl Crombine and traveled to Sri Lanka with him on a collecting trip. She was a prominent member of the Department of Entomology at the Smithsonian for a number of years during the 70s and 80s. Beth had a particular interest in bees and practiced a positive attitude among activities at the museum. She will be missed. Thank you.
Thank you very much.
Sure.
Moving now to officer reports. Do we have reports in here or on Zoom from officers? No. Okay. Introduction of new members, which is also an office report from Matt Buffington, our membership secretary, who is not here today, but he gave me the list of people who applied for being new members. They are Louis Nastasi, Angela Sands, Gina Angeletta, and Mark Athride. So welcome. Do we have here any new members or visitors that would like to introduce themselves? Never. Thank you very much and welcome. Do we have anyone on Zoom that would like to introduce themselves?
I can introduce myself. My name is Angela Saenz. I'm a PhD student at the University of Maryland in the Entomology department. And I'm working with Dan Gruner. He's my advisor, and I'm working with invasive forest pest and biocontrol. So thank you for having me.
Thank you. And thank you for becoming a member. Welcome. Okay. I don't remember us having any unfinished business. Tell me if I'm wrong. Nope. Any new business that somebody would like to introduce to us now? I don't have new business either. Okay. Let's move to presentations of notes. Does anybody have show and tell? Yay. Can you please come closer to the microphone?
Sure.
Yeah, here it's perfect because they can see you and hear you.
Okay.
And then I'll take a picture after.
Yeah. So actually, I started this when I was president last century. But recently, well, not so recently, we received a collection of ants. I thought it was appropriate. In a couple of parts from a guy named Arnold Van Pelt, who was a professor at Greensboro, several places, but Greensboro College in North Carolina. And he gave his collection to us. He worked mostly on ants at several institutions. And when he arrived at Greensboro, he had one big project with Big Bend National Park. And we, in 2013, I think it was, 2014, Boyd Shockey and I were called by his father down to their house and picked up the rest of his collection and his archives. I worked with our archivists to get them all arranged. So we got field books, correspondence, everything. And then while I was away last year, I came back to the sorting center with several boxes of things from Van Pelt that apparently contained specimens that never made it to Schultz's lab. So recently, when I had a lull by helping McCoy, I went through all these and I spread them all out, and they were all, almost everything, 90% was Big Ben project. So I was going through these cartons and spreading out the vials, and they all have numbers inside. And then I found one. Can you hear me okay? I think now. Okay. Then I found a little box, and I opened it up, It contains 11 vials collected between 1948 and 1950 by E.O. Wilson. We used to give him a class one, which I thought was quite interesting. So some of them are still have alcohol, some of them don't. They're mostly from Alabama, and Ed was from Alabama originally. He entered grad school at the Museum of Comparative Zoology at Harvard in the early 50s, mentored by Bill Brown, another colleague of mine. But these were collected before he went to Harvard. So these were collected from his homeland in Alabama, and one vial is from Great Falls. Some of you probably know that he's famous for having collected Rock Creek Park when he was 10 years old. But anyway, it was kind of an interesting find. And so I segregated these out. I've given them to all of this that I spread out into reasonable collection containers to Eugenia for Ted and Sean. Sean and Ted were very surprised when I told them about this. So anyway, I'll put these vials over here. These all, it's clearly mentioned that he collected them. He identified some of them. And it seems appropriate for tonight. So.
Thank you very much. That sounds fascinating. Does anyone else has show? Please come.
I brought here a new volume of the moths of North America. It's right off the press. It was late last year and just received by myself. If you don't know the series, it started years ago in South Carolina, and they've done, gosh, I don't know how many volumes, maybe 25 now, of moth families and parts of moth families. This one is very new and done by Quinter and McBride. Very well done. Their formatting is just excellent. They've got illustrations of the genitalia, the field collection sites, the moth adults, and the immatures. It's just fabulous to see. I think I'll just put it over here on the table and you can look at it later. It's rather bulky to send around. But the Mona series still goes. I'm sure there's other manuscripts being finished off as we speak.
It was started by a guy named Richard Dominic.
Yes.
Started by Wedge Foundation.
Yeah.
Several members of our department worked with Jack Brown. So,
Ron Hodges.
yeah, Ron Hodges. He went to Congress to try to get an allocation for the project. And then it was.
Thank you very much. Okay. Program Chair Renan Carreño, can you please introduce tonight's speaker?
All right. Tonight's speaker is Tais Ribeiro. Tais is originally from Brazil, where she completed her master's degree before moving to the United States to pursue her PhD at University of Maryland. And she currently works as a lab coordinator at the UMD and is part-time lecturer at Montgomery College. She also is a volunteer here with us at Smithsonian. She's working on Lepidoptera. So tonight she'll be presenting a talk titled Biotic and Abiotic Drivers of Diversification in the Oil Collecting Bee genus Chalepogenus. Please.
All right, thank you. Thank you, Renan, for the introduction, and thanks everybody for being here. So today I'm going to talk about Chalepogenus bees, so these cute little bees that we have here. And this is my the research that I did for my PhD. So I'm gonna go through that, like what did I study with them. So the way that this presentation is structured, so I give you like a quick background, and then I'll tell you like what were my main questions in my research, and I'll go through each of the chapters that I had for that. So one of the greatest puzzles and one thing that many of us are interested in is biodiversity. So we want to know what is the biodiversity out there, where are the species distributed, when do they originated, And there are many fields in science that focus on that. So we have taxonomy, that is like focusing on understanding what are the species. We have the study of phylogenetics, so studying what is the how those two species are related to each other. And we have the study of evolutionary processes, right? So then to understand how those relationships came to be. And so all of this together is in systematics, which is a field that many of us here are interested in and work on. And really the study of evolutionary processes is what guides also the questions and that we can answer like from all of these other fields. So and then evolution is driven by both biotic and abiotic factors, right? So we have interspecific interactions, which are biotic factors that can drive the evolution that can act as selective pressures influencing the species. And we have abiotic factors such as the biogeography, so where the species are located, and then what was the history of the areas that they're in that they can affect their evolution, and also the climatic variation, right? So what is the history of the areas that they are, like what is the climatic history and how does that can change patterns that can lead to the evolution of species. And so all of these together, in my research, I study how all of these different factors can be affecting evolution. And I did that by focusing on bees from the genus Chalepogenus. And so Chalepogenus are in the tribe Tapinotaspidini, which is in the Apidae family. And there are 29 known species, including some undescribed species also. And those species are separated in morphological groups, which I will refer to in this presentation also, and I'll call them as morphogroups. And they are in South America, and they are widely distributed in South America. And what's interesting also about them is that they are oil collecting bees. So these bees, they have specialized pollination interactions that they collect floral oil rewards from specific flowers. So even though they visit other flowers to collect the pollen and the nectar, they have those specific relationships with the plants that they collect their floral oils. And they use those floral oils to feed their larvae and waterproof the brood cells. And that leads to interesting ecological patterns. In Chalepogenus, we have the variation in the ranges, which leads to the variation in climate, and also these variations with the floral hosts that they have. And all of this together makes Chalepogenus a good system to study this, how those different processes, how those different factors can be driving evolution. And then like thinking about that can be applicable also for like other taxa, like this type of thinking. And however, we didn't know what explains the evolutionary history of the group given those different traits. And the evolutionary relationships, the phylogenetics and the systematics of the group was unclear. So what was known about that? So in the taxonomic revision of the group in 1999, that's when Roy Galstina separated the species into those morphogroups that are here, and then he proposed some relationships between those different groups of species. But then more recently, in some molecular phylogenetics, there was a pyrophily that was recovered in the group with some of these species. So here in purple, and the species Godianus and Parvus that are in those groups, as being in a different clade, separate from group Millery, which is the one that contained the type species. So then that they would recover a paraphily in this group. However, in these works, many of the Chalepogenus species were still missing, so we didn't really know what was going on with the other species. So we needed to like investigate these evolutionary relationships by adding more taxa. So in my research, I asked, what are the evolutionary relationships in Chalepogenus and what are the drivers of evolution? So how are those different factors affecting the evolution of the group? And then to do that, I did 3 chapters. So I developed evolutionary hypothesis, and then I investigated the evolutionary relationships, and then I looked into the drivers of evolution. So first, for developing evolutionary hypothesis, what I did, so we can think of how the natural history of the group is reflecting the evolution of the species. So what we can see of where the species are located is a reflection of their evolutionary history. So using natural history data, so the locations and then all the ecological data of the species, we could infer evolutionary hypotheses. So we can think of like how those species are related based on their ecology. And that is an interesting thing to do, like especially in taxa that have ecological data available, but didn't have a phylogeny yet, so that we can actually look into the phylogenetic relationships in the group. So to do this, I did a literature review, and then I identified the ranges of the genus, the climatic preferences, and floral host choices, and then I integrated all of these traits to think of ecoevolutionary hypotheses. So first, to identify the ranges, I compiled georeference observations from databases and from literature, and then I evaluated these ranges based on biogeographic subregions that are already published for the neotropical region for South America. And then for the climatic preferences, I use those same geographic observations, georeference observations, and then couple that with current temperature and precipitation variables, and did a principal component analysis to compare this data. Then to identify the floral host choices, I gather floral visitor data from literature, and I focused mostly on B morphological traits. I focus on the tibial hair and thoracic hair, and also on the body size, like so just the size of the bees and the wing length. And then I integrated all of these traits. So to do that, I did a multiple factor analysis, and in this type of analysis, we can integrate both categorical and quantitative variables, and then the traits will define clusters with this different with where the different data is in the multivariate space, and then we can evaluate how this data is clustered to compare the role of the different variables. So, for example, if we had some data that in the graph will look like this, so we had the teal triangles and the purple diamonds, for example, you see that they're overlapping in some way. So then we could think that the groups that are more that have like more similar characteristics in this multivariate space could be more closely related, or could have more phylogenetic similarities. And so we can think of representing those relationships like this, right? Like with those two groups are more close than the other ones. Okay, so what did I get for this first chapter? So what were the results? So first for the ranges, we compiled 1991 georeference observations, and most of the data was from databases and from museums, from which 670 of these coordinates were unique. And then we plotted that in so we see the ranges in South America. So here, those different points and the different colors represent the groups of species. So each one is a group of species. And then we see that the morpho groups are mostly clustered spatially. So we see that there are some groups that are in the South Andes, some that are in the North Andes, and some that are non-Andean and related to grasslands in South America. And when we looked into the biogeographic regions and using the Mahoney biogeographic regions, we see that those are corresponding in the with some of those subregions. So we have like the South Andean ones are in these specific regions, and then the ones in the North Andes are in the transition zone, and then some in the Brazilian subregion, and then the non-Andean are mostly on the Chacoan subregion, which is associated with the grasslands. So we have the Pampa in South America, most in Chaco. And yeah, and then like these are the other groups that are in the paraphyletic, so that's why I left them separate, but they're also in the Chacoan subregion and overlapping with some of those groups. So now for looking into the climatic preferences, so I did the PCA, and then here we have those different variables that are representing those variables, and then the contributions of each of those variables. So those are the bioclimatic variables. And we see that there is a larger contribution of seasonality in here. So then we have those different variables here, like when we're comparing PC1 and PC2. So we see here that PC1 versus PC2 explains 50% of the variation that we are seeing. And then that the variables related to precipitation were more important, like had like a higher contribution here. So yeah, so like related with precipitation and seasonality, which indicates that either rainy summers or rainy winters are being are contributing to the variation that we see in the data. And then for PC1, and then for PC2, we see that precipitation of the warmest quarter and of the coldest quarter are contributing more to that axis. So that like either like, yeah, the rainy summers or rainy winters are related to that. Yeah, so then then we could maybe call those quarters like that. And then when we plot the data, so here again, like this is the different colors of the different morphological groups. And what we see here is that there are two main clusters. So there is one cluster that is with the groups that are in the Andes, so the red and the orange ones, which is group Cerulus and Herbstii. And then all of the other groups separated than the other ones. So yeah, so that's when we compare PC1 and PC2. And then when we look at PC1 versus PC3, we see that the most important variables for PC3 were isothermality and temperature seasonality. So isothermality is a metric of the variation of the daily temperatures in relation to the annual variation. So we have temperature playing a role like when it comes to PC3. And what we see here in the data is that everything kind of clusters together, but then we have this group, the yellow one, the group Fogeli, that differs from the other ones due to the isothermality. So it looks like it's adapted to like less annual variation to of those temperatures. And now when we look into the floral host choices, we wanted to identify like what are the preferences. So compiling all this data, here we have in the boxes here are the groups. So with the species in each group inside, and then the colors refer to the plants, to the plant genera that they visit. And the bars just show like the number of observations that we had for each one of them. And then what we see is that there are some different levels. So most of the species, most of the groups, the species inside a group have the same plant that they visit. So we have many groups that visit mostly Calceolaria plants in orange here, and then we have one group which is the one that contains the type species again, and that one is more generalist. So we visit plants from different different genera, but two genera in the same family. So Cipella and Cicericium, which are in Iridaceae family. And then we have other groups that visit Nierenbergia, which is a different plant in the different families, but they're also like very specialized in collecting the floral oils in these plants. So we see those different adaptations that the groups are still structured from like the plants that they visit. And then now when we put all of this together to integrate that, so we see that so this is the results of the multiple factor analysis of the MFA, and then it's similar to the results of the PCA in that like there we have the two axes, which are the dimensions 1 and 2, and the arrows with the different variables that were included, the different groups of variables, and then when we plot the data here, we see again that there are two main clusters. So groups Herbaceans to reduce, so the orange and the red one are overlapping, and all the other groups have some sort of overlapping, but isolated from the other ones. And group Millery, which is the type 1, is somewhat isolated. Yeah, and then just like showing like the separations here. But then when we look into dimension 3, we see some more separation there, so that we see groups Herbaceans and Cerulus again together, and all the other groups, most of the other groups together, and then group Fogale, the yellow one, isolated from the others. So, and that might be because of the geographic location of this group. So group Fogale is located both in the North Andes and also in the Chaco. So then that separation might have led to the separation with the other ones, from the other ones. So then in general, when we put all of this together, we looked at some of those groups have similar patterns. So we could think of these relationships between the groups based on the plants that they visit. So here we have like the different plants that they visit. So Cerulus, Herbacean, Fogale visit Calceolaria plants, so the yellow plant, and Millery is the one that is more generalist, and Agrippas visits Nierenbergia, so the purple one. And they have similar patterns also in the geography, right? So then so we have Agrippas and Millery that are more in the Chaco region here. These two are Andean, and then this one is in two places, right? So then it's mostly like in here, like following the North Andes, and then this area. So if we think of these, we can think of these relationships here, and if we're grounding on the biogeography, we could think of the relationships between those different groups. So we can think of evolutionary hypotheses of how those groups can be related, and then thinking of the biogeography because it looks like it's what is driving more these relationships. And so to do that, like I disregarded the groups Geogenus and Parvus, right? Because there were the ones that in past phylogenies that were recovered as paraphyletic. So just to focus on this one group. And then we can come up with thoughts, so evolutionary hypotheses that we can think of. So I summarized them in east to west to east, and central origin. So like thinking of an east to west hypothesis, we could think of an ancestor of the group having an origin in the grasslands, and then moving to the Andean region. So we could think of a generalist ancestor adapted to more humid climates. But then if we would think of the biogeographic history of the area, we know that that there was Andean uplift that happened around 15 million years ago. So we could think that if that would be the case, then there will have transition, like the group will have to be older than 15 million years ago so that it would be able to have this transition. Now, if we think of a west to east hypothesis, we could think of the ancestors being specialized in Calceolaria, for example, like the yellow plant here, and then migrating to the grasslands, from the Andes to the grasslands. But then again, like they will have to be older than 15 million years ago. And we can also think of a central origin hypothesis with an ancestor that will be more in the central region here, and then it could have expanded both to both sides, so to the east and the west, and that could have happened around 10 million years ago. Because that's when there was grassland expansion in South America, and then they could have followed that pattern of grasslands expansion to expand there. So we see that the ecological preferences are reflecting the current taxonomy of the group, and then we can think of those different hypotheses. But of course, we can't really test this hypothesis if we don't have the phylogeny of the group. And all these hypotheses would estimate the age older than 10 million years ago, thinking of the biogeographic history of the area. But okay, we needed to look into the actual phylogeny, right, to see to like hypothesize again, like what could be happening. So to look into these evolutionary relationships, and again, like just to remind you, so we had the current phylogenetic hypothesis that the group was non-monophyletic, right? So then they had those groups that were separated, but then it was still missing in past molecular studies. They needed more sampling to like really look into the whole group. So that's what I did. So I had a large, so for inferring a phylogeny, right, we need the sampling, we need the characters, and we need to do phylogenetic methods to build a phylogenetic tree. So in my case, I had a sampling of Chalepogenus, including like more species. And I use UC markers to build phylogenetic build phylogenetic trees using different methods. So I had this larger sampling that included 16 out of 21 of the described species, and I also included undescribed new species with a total of 96 specimens in my data set. And I use as outgroups species from Lanthanomelissa, which was recovered before as a sister group to some of the Chalepogenus, so to the ones that are paraphyletic. And also included published outgroups from the whole tribe, Tapinotaspidini, and some of the sister tribe. And just to note again that most of the specimens that I use are from museum specimens. So we had some of the field collected, but most of those specimens were from museums, so ones from the museums. And then we use UCE. So UCEs are highly conserved parts of the genome that can be used to investigate those relationships. And they have a highly variable flanking region that we can use to look into the relationships between the genera. And I use the bait set that was built for Hymenoptera. And our main data set ended up having 1997 total loci. And so for building the tree, so I use different methods. So I use both concatenation methods, so Bayesian and maximum likelihood. And I also use coalescent methods to make gene trees. And so I use Astral and TreeQMC. So we could look at different ways of building the trees. So what happened? Like, what was our result? So with that, we were able to recover a well-supported phylogenetic trees. So again, here the colors are the morphological groups, and I'm going to zoom in on some of them. But those are the morphological groups, and then the specimens in different species that we had here. And so then here's the Bayesian tree as an example. But then we generally found a similar topology for the other phylogenetic methods. And then what we have here is that we see that there were two main clades. So there was one clade that is the one. So we also recovered the genus as paraphyletic, as non-monophyletic with that clade, with those species in a different clade. And then we have clade 2 in clade 1, and then we have clade 2 showing that the genus was not monophyletic, but the morphogroups that were described in the taxonomic revision, those morphogroups were recovered as monophyletic. So we see that the blocks of color are together. So first in clade 1, that was the clade that has the groups Geogenus and Parvus, which is the other group, and they are sister to Lanthanomelissa. So they are sister to a different genus in there, and there are also other Tapinotaspidini here that are separating these groups. So based on that and based on previous molecular phylogenies that that we published and that included Lanthanomelissa, we can say that these species are not in Chalepogenus anymore. So there is like a taxonomic revision that is needed for this group. So then that's in clade 1. And then for clade 2, what we have here, so we have two subclades here. So we have clade 2A, and that includes group Millery, which has the type, sister to most of the new species. So the new species, undescribed species, are likely Chalepogenus because they are in the same, they are sister to the group Millery. And sister to that clade, we have all the remaining groups. So that we have all the other groups in a clade sister to that clade that includes the Millery and the new species. So in conclusion to that part, we see that Chalepogenus is not monophyletic, but the groups and species are monophyletic. There's still some taxonomic revision needed, but now that we have this phylogeny, we can construct evolutionary analysis. So now it's time to look into the different drivers of evolution. So I could do different analysis to look at the evolution. So to look into that, the question here was to understand what could be driving the evolution in this group, right? So we see that we know that ecological traits can drive evolution, and we can test that using the phylogeny, right? And then so would it be what is the role of biogeography, of the climatic variation, and of the interspecific interactions in driving that? So to look into that, I use the phylogeny to infer the biogeographic history, to understand how the climatic preferences evolved in the phylogeny and the evolution of the floral host choice. And also to test the hypothesis that we came up with in chapter 1. So to do that, I first needed to estimate the ages in the tree that I had, and also to measure diversification. So to do that, I built a smaller tree using the 200 most informative loci, and then I built a tree using Beast, and I dated it in using 4 calibration points, using the dates from previous works in Tapinotaspidini, so in the tribe, and also in the outgroups. To do secondary calibration for dating this tree. And then I also pruned the tree to 1 tip per species. Yeah, and then for measuring diversification rates, so diversification is a metric of evolution, which is characterized by the difference between speciation and extinction rates. And one way of visualizing that is to plot the number of lineages that are evolving through time. So the lineages through time plot. And then we can fit several models to estimate diversification rates based on those rates of speciation and extinction. So, and then I did some other, so I fit different models to look into the diversification of the group. Yeah, so then after doing that, then I needed to estimate the models of trait evolution for each of those different traits, and reconstruct the ancestor state so that we could see, we could estimate what was the evolutionary history. So first for the biogeography, what I did is that I used biogeobears, and then I used all the models that they had available. So GC, Bayer-alike, and Diva-alike, both with and without the jump dispersal parameter. And these models, they use the current ranges and the phylogeny to estimate how those ranges could have been reached over time. So, and then so we needed the models, and then I used the prune time tree that I built. And then the areas that I used, so I used the 7 areas from based on the ecoregions from WWF. So based on biomes that are in South America. So now for the climatic variation, what I use this method called bfind to reconstruct the climatic niches being formed by the phylogeny. So the way that it works is that it estimates those niches based on the optimum and the breadth. So it characterizes those niches like that. And also, again, using current observations. And then we use I looked into the I looked into the temperature and the precipitation in this group. So yeah, it estimates the climatic optimum, which is what is the preferred temperature and precipitation for the species, and the breadth, which is the range of the values that the species can still exist. And then we built the stochastic character mapping to map the evolution, to do the ancestor state reconstruction in the tree. Yeah. So again, for that, like I needed the prune time tree, and then I needed the occurrence data that I had, and then the variables that I used were the mean annual air temperature and annual precipitation to look into how those variables have been evolving in the group. Yeah, and then for the answer-specific interactions, so for the host choices, so again, like it's using different models. So I estimated the transition rates using core HMM, and then I look into the character-dependent models, like in how those different preferences evolved. So again, I use the same prune time tree, and then here I looked into the different plant families that those species or that the bees are specialized in. All right, and then what do we get from that? So, so first for looking into the time tree and the evolution. So here we have the time tree. So it's a smaller data set than the one that I had from the chapter 2, but then it still had a similar topology. And so we had the clade 1 and clade 2 being recovered here again. But then what we had here, so we estimated the origin of the group at around 34 million years ago of the ancestor of this whole group. And the main clades were around 30 to 26 million years ago. So we have clade 1 around 26 million years ago, and then clade 2 around 30 million years ago, and clade 2A around 26, and clade 2B around 16 million years ago. So clade 2B is the youngest than the other ones, around 16 million years ago. And now when we look into the diversification rate, so here we have like the different diversification rates. So the blue color means higher rates, so higher rates of diversification. And then here in the tree, so that's the mapping. So then when we see like the different colors, so that's the rates that we are recovering like across time in the phylogeny. So we see that there are mostly high rates closer to the root, so darker colors in there, and in several of the tips. And when we put the lineages through time plot, so here we have the number of lineages in the y-axis, and here the time. So we see that there is an exponential growth. So lots of those, so especially at this time around 16 million years ago. So we see that there is this very fast growth in the amount of lineages that we have. So that could indicate that the species are still diversifying. So the group is fairly new in this sense, and then there's still diversification going on. And now when we look into the biogeography, so here we'll go over the ancestral range reconstruction. So what we have here, that we have the map, and then those different colors are combinations of these areas. So it doesn't reflect exactly the same colors here because it's a combination of colors. And then here at the bottom, we have the temperature variation over time. So here the time, and then the historic variation. And yeah, and then I'll show you the tree with the pie charts that are the likelihoods of the ancestral ranges. So here we had it, and then we had all these different pies with those combinations of areas as ancestral ranges. And then we used the best model selected was the Bay Area like. And yeah, so then like let's go clade by clade. So first, with the ancestor of the whole group, we see that again, like that was around 30 million years ago, and then that was in an area of combination of Atlantic forests and grasslands, and that could be related, and then that relationship could be related with a cooling event that happened around that time, and this cooling event could have led into a transition from forests to savannas in the group. So later on, like around 30 to 26 million years ago, we see in clade 1 that there was a switch. So In clade 1, the range contracted, and then they stayed more in the Atlantic, like Atlantic forests and grasslands, but not in the temperate grasslands to the south. So the yellow is not present for that group. But then when we go to clade 2, we see different. So we see for clade 2, in clade 2A, that is an expansion to they stay in the grasslands, but they go away from the Atlantic forest side. Now later on, like with time, and then now when we get to the Miocene around 16 to 14 million years ago, that was when there was the thermal maximum. So the temperatures got higher in the world, but also in South America. And then at the same time, there were Andean shifts going on. So the Andean uplift happening in South America. And so this uplift and then this climatic optimum, these changes in the temperature could have led in clade 1 an expansion back to the temperate grasslands. And in clade 2, a contraction to stay only in the savannas. And but in clade 2B, that's when we see the expansion to the Andean region. So they expand into the central Andes. In this clade, and that expansion stays. That expansion also happens later on in clade 1, around 8 to 6 million years ago, related to the grassland expansion that happened around that time also. So and then at the same time, also in clade 2, so then we see an expansion to the savannas. And another expansion to the South Andes in some of those groups around that time. So what all of this means is that the switches that we are seeing in the areas that those bees are occurring, and then the areas of the ancestor is congruent with events, with bioclimatic, biogeographical events that were happening in South America. So the Andes expansion, and then the temperature changes and everything. So they're all connected. So we see here that the group has the origin in the grasslands and the forest area, and then later dispersal into the Andes. And then those timing of events were congruent with this geological and climatic history of the area. And now when we went to look into the climate, so here we have in the center, we have those violent plots that showing the variation for the temperature optimum in this side, and then breadth on the other side. And here we have the different the tips that we have, like, so the different taxa. And then what we see here is that there is an inverse relationship between the optimum, the temperature optimum, and the breadth for pretty much all the clades. So generally we see, for example, in clade 2B here on the top, we see that it has a lower temperature optimum than the other ones, but it has a higher breadth. So even though it is adapted to lower temperatures, but it tolerates a higher variation of those temperatures. And then so when we look into the phylogeny, so here the colors are the temperature that is that they are so the value. So in this side is the temperature optimum, so the red color means higher temperature and blue means lower temperature. And then what we see here, the ancestor had an intermediate temperature when compared to the other ones, and breadth. And then in clade 1, we see that they are adapted. The ancestors were adapted to a higher optimum but a lower breadth, so higher temperature but less variation. And then in clade 2A was similar, so also higher temperature but smaller breadth. And in clade 2B, that was the opposite. So clade 2B again is the one in the Andes. So that's the one that we see that they were adapted to lower temperatures, but higher variation of these temperatures. And then when we looked into the precipitation, And it was more or less similar. So we have in clade 2B again, like they tolerate a lower precipitation. And in clade 1 and 2A, there was not a big change between these between those preferences for the ancestors. Yeah, but then there was in clade 2B, like there was this lower precipitation, but also lower breadth. So here, like the darker blue means lower, right? So then it's lower precipitation and a larger breadth of like larger variation of the amount of precipitation that they can tolerate. So we see that there were variations in the climatic niches by clade, and there was this trade-off between the niche breadth and the optimum in the temperature. And we see that the Andean clades, they are adapted to colder and drier conditions in comparison to the other groups. Now we're looking into the interspecific interactions. We did the ancestral state reconstruction to look into the current plant. So here we have those squares are the current plant preferences. So the different colors are the different plant families again. And then the clades. And then here in the tree, we have the pies showing the likelihood of the ancestors. And then what do we have here? So we have that the ancestors was probably adapted to Malpighiaceae, which is it's actually the older oil offering plant family. That none of the current species actually visit this plant. Like most of the described species don't visit this plant. But yeah, and then the ancestor was recovered to be Malpighiaceae. And in clade 1, we have a switch to Solanaceae around 16 to 14 million years ago. So again, the time of the Miocene climatic option, so everything changes around that time. So it also changes the plant preferences that they have. And in clade 2, in clade 2A, they become more generally, so they visit mostly Plantagenaceae, but also some Iridaceae in yellow. And then in clade 2B, the Andean one, they visit Calceolaria. So they visit a completely different family of plants. And later on, around 9 million years ago, we see like a switch to Iridaceae. So there was a switch that happened later on in both in clade 1 and in clade 2A. And around 8 million years ago, we see a switch to Solanaceae in clade 2B to for one group in clade 2 groups in clade 2B, and then one group in clade 1. But what is interesting about that is that all of these host switches, so we were looking at the plant families, right? But those host switches are actually matching the age that is recovered for the plant genera that they currently visit. So yeah, and then here just like in detail, like when those different genera were originated. So what that could be telling us is that the plant genera that they are visiting are younger than the bees. So the plants that they are visiting now were not even around when the species, for the ancestor of the species. So like when these plants originated, and that we see a switch from the bees to like start visiting those plants. So again, for this part, we see that the ancestor was Malpighiaceae, that the host switches are congruent with the origin of those new plant genera, and that the ancestors of the bees is older than the ancestors of the current plant genera that they have now. So in conclusion, for that chapter, so if we go back to the aims, so when we're looking to the evolution, what do we have is that we see that the origin of the group was around 34 million years ago, but then most of the diversification happened around in the last 15 million years ago, in the last 15 million years. And when we look into the biogeography, we see a very strong biogeographic effect in the evolution of the group. And when we look into the climatic variation, we see that those climatic transitions could be connected to biogeographic events. And again, for the interspecific interactions, that the current host plants genera are likely not ancestral. But like putting it all together, we also see that all of these ecological transitions are associated with similar biogeographic events. So to put it all pretty much in one figure, so when we look into the history of the group, we could think of the group having an origin in the east, and then with group Millery here, and in more preferring Iridaceae or being more generalist, and then there were expansions to the Andes later on. To the Andes and the grassland. And then if we think again of the hypotheses that we had in chapter 1, we see that none of those hypotheses were actually supported. So none of them are like the same of what we saw when doing the analysis, when testing it. But the east to west is more congruent with the results that we had. So in general conclusions, we wanted to see what were the evolutionary relationships with Chalepogenus. So we see that Chalepogenus is non-monophyletic, and then there are at least those two clades, and that originated around 34 million years ago. And then most of the diversification again was in the last 15 million years ago, but then there are still diversification going on. And looking into the drivers, we see that biogeography and climate are likely important drivers, but the plant preference not so much, right? Because the genera had a different are younger than the bees. So next in this research from Joy is looking into other analysis of diversification and looking into this evolutionary analysis. And also there's a taxonomic revision necessary for the group. So with that, I wanted to thank everybody. Thanks for having me. And I wanted to thank my dissertation committee, my advisor, the lab, and also the museums and personal collections that lent us the material so that we could do our work. And yeah, and then again, like my whole work like wouldn't be possible without museum collections and natural history data. So it's all based on that. And yeah, and thank you so much for you all to be here. And yeah, thank you. Obrigada. Happy to answer questions. Yeah.
Great talk as well.
Thank you.
I'm sorry if I missed this, but could you just say a few words about like what are they doing with the oil that they're collecting?
Yeah, so they mix with the pollen and give it to the larvae, and they use it also to waterproof their brood cells. But there's still some work being done. Like so, there are different oil collecting bees. So these bees, like the tribe Tapinotaspidini, like all of them are oil collecting, but there are also other species of bees in other families. So like in Melitidae, for example, a different family, that there are also like some bees that are oil collecting. And it's really cool, like the things that they do with it. Yeah, but it's mostly to waterproof their brood cells. Yeah.
I'm curious, maybe this is a bit from the wrong direction for botany, but if it could be actually the other way around, instead of the plants driving the evolution of the bees, if they're younger, if the plants are actually being driven by the bees.
Yeah, exactly. So then that's why, yeah, we thought that it would be like, oh, yeah, the plants are going to be driving, but since the genera that they visit now are actually younger, so then it could be the other way around, right? Like it could be that those plants are evolving to offer those oils with the presence of oil collecting bees around, yeah, like with the other ones. So yeah, so the plants that they produce those oils, they don't offer nectar. So they choose to, like there is the switch so that they can offer oils instead of the nectar. But yeah.
And then one more question, which was there any difficulties with using mostly museum specimens in a very genome heavy project?
No, so with the UCEs it was actually fine, which was good. Yay. So a lot of the specimens that we had were like collected and they were like frozen, but then yeah, like for the museum ones we had we got a leg and then we used the UC. UCs are
These are pretty sizable. So.
What the
specimens are working with, so like a leg will go pretty far.
Yeah. I mean, yeah, when we would test the amount of DNA, it was like mostly like below what they wanted for the protocols, but it worked out, so that's good. Yeah.
Yeah, thanks for the talk. Can you say something about clarifying of the groups that were close to what was called Latano?
Yeah.
Which way around this other like one or yeah, what's causing the paraphylegy?
Yeah, so actually when looking at the morphology, like I'm just going back to the thing. When looking at the morphology, they are also a little different. So for example, oh my God, I have a lot of slides, sorry. Here. So we got the purple ones, right? In the bottom there. Here. So here we have like the Gaudianos and Parvus. And Parvus actually really looks like a Lantana melissa because they are tiny and they're very yellow, like the Lantana melissas are. And they also have 2 submarginal cells in the wings, and all the other ones have 3 submarginal cells, but Parvus has 2. So Parvus really looks also like a Lantana melissa. But yeah, and then when we do the molecular analysis, it also shows up as sisters. So we're proposing like changing Chalepogenus parvus to Lantana melissa. And these are the ones, so here we have Gaudianus and Nephi. And those, previously they were described as a different genus. So they were described as Lantanella, which is a genus that doesn't exist anymore, but should exist again because that's what those probably are, you know, like those are different from the Chalepogenus. So there is also some morphological evidence for these switches, for the differences. Yeah.
So are you proposing classification changes?
I didn't in my work. I didn't like explicitly say. In my work, in my master's, I worked with Lantana melissa. So I worked with just this clade, but not a lot of Chalepogenus in there. And then in there, like we proposed that that change, but we didn't change yet for all that. Yeah. So yeah, I didn't describe or. Oh yeah, so that's the thing with this new species. They are not even in the same areas that we're looking. So these new species here, like these here, they are in the Cerrado. So like in different areas in Brazil that there's not enough collections for them. Yeah, like there's not enough material being collected. So probably there's a lot of other Chalepogenus around that. Yeah, so we have it in Cerrado and Caatinga. And then I didn't even include here because all of the other ones that are described are not even there. So yeah. And then, you know, like I asked, like when I was doing getting all the data, so I asked people in many museums in South America also like to see, and then many of them just said, hey, we have stuff where we don't know what it is, you know, like it's not even ID. So maybe if I would go there and try to ID some of the stuff, that would be more data buried. So yeah, we got what we could get. Yeah. Well, it still exists around here, so you can always ask me later. But
Yes.
yeah. Well, thank you so much. Thanks for having me.
Well, and thank you very much for everybody here and on Zoom. Can I have a motion to adjourn the meeting? Second, all in favor?
Aye.
The meeting is adjourned. Thank you.